Okay, awesome. So the lecture objectives we're going to start off with is describing how the urinary excretion of solutes and water influences the volume and composition of plasma. So basically how the fine tuning of urine, we'll talk a lot about this throughout today and the rest of the week, but the fine tuning of urine also affects the composition of plasma in terms of the water content, in terms of the solute content, so sodium, potassium, chloride, etc. We're also going to talk about how the basic renal exchange processes occur. So that's filtration, secretion, and reabsorption. We'll talk about some of the laws that influence that exchange process. And you should be familiar with those laws. So the Starling's forces, you would have looked at that as you talked about capillaries and the movement of plasma across the capillary beds. So we're going to call on some of that information again. We're going to define the following terms. So we'll talk about filtered load, glomerular filtration rate, or GFR. We'll talk about things like clearance, transport maximum, and renal threshold as it applies to those renal exchange processes. And then finally, we'll talk about micturition. So micturition is the process of urinating, and we'll talk about the various branches of the nervous system that influence and control micturition. Okay, so let's start off with water balance. So let's start off by talking about some of the functions of the kidneys. So we know that we've got kidneys, right? They're paired organs. They're retroperitoneal organs, meaning that they lie behind or posterior to the peritoneum, so really close to the posterior abdominal wall. We know that the kidneys are in charge of filtering the plasma, which is basically kind of cleaning the blood and fine-tuning that filtrate to produce urine, which is our excretory product, and that can leave the body. That's a waste product. So a lot of what we're going to speak about is what happens after we filter that plasma. So we filter blood, and then what happens? What are the processes that are involved where that filtered blood then becomes urine and can be eliminated from the body? Let's talk about some of the other functions of the kidneys. So they regulate the plasma ionic composition. So basically dictate how much sodium, potassium chloride is in our plasma. That's really important in terms of our blood pressure. It's also going to regulate our plasma volume, also important for blood pressure. That's going to tell how much water is in our plasma, which is a really important function of our blood volume and blood pressure. It's going to help regulate our plasma osmolarity. And when I say the word osmolarity, I want you to think about the salt level or the solute concentration. So another word for osmolarity is, what is the saltiness of this this fluid or this substance that we're talking about. And so the kidneys also help to regulate the amount of sodium level in our plasma. The kidneys regulate plasma pH, so really important in terms of balancing our pH, and they do so by maintaining bicarbonate levels. We'll talk about that later on, and also the way in which they clear hydrogen ions. We'll talk about that as well. The kidneys remove metabolic waste products and foreign substances, so medication, toxins that we may ingest are cleared or eliminated through the kidneys. And then finally, other functions are kind of secondary functions here. They secrete erythropoietin and renin, which are hormones. Erythropoietin is going to be a precursor to the production of brand new erythrocytes or new red blood cells. Renin is going to be a really important hormone in terms of balancing our osmolarity. We'll talk a lot about renin. It activates vitamin D3 to calcitriol. So when we are exposed to sunlight, this is a natural way of us taking in vitamin D, but it needs to be activated or converted into its bioavailable or active form, and the kidneys helps to do that. So when we're exposed to sun, that sun can be taken into our body in the form of vitamin D3, and then that is activated to its bioavailable form. And then finally, gluconeogenesis, which is another process that we'll talk about here, but also in the GI system, also is done by the kidneys. Now, in terms of the overall anatomy, and again, I won't spend too much time on the anatomy here because we're going to go delve into this more in lab, but let's look at what a kidney looks like here on the right. So this is a kidney. This is a frontal or kind of a midsection cut of a kidney. And right away, we can see two distinct regions of the kidney. To the gross eye, there's this outer pale staining region called the cortex. And that is different from the inner, darker or deeper staining region that's called the medulla. So this outer region is the renal cortex. This inner region is the renal medulla. And as a part of the renal medulla, we have these renal pyramids, right? So these congregates within the medulla are called renal pyramids. Now, these two different areas of the kidney is where we find two different parts of the nephron. So the nephron is this partly tubular, partly corpuscle structure that's shown here in yellow, and it spans from the cortex. We'll talk about the parts of the nephron that are in the cortex, all the way down into the medulla. We'll talk about this part of the nephron that goes down into the medulla, but we want to understand this kind of polarity of the nephron. There's a cortical region of the nephron and a medullary region to the nephron as well. So we're going to bear this in mind in terms of the general anatomy of the kidney, and then we're going to go a little bit further and talk a lot about how the nephron looks, so the anatomy of the nephron. I also want to point out here the renal pelvis, which leads to the ureter. And the ureters are where that final product, when it becomes urine, can drain down into the bladder where it's stored until it's time to be eliminated when we void the bladder. And then lastly, I want to point out here the vasculature to the kidney. We've got the main vessel, which is the renal artery, and then the main vein here, which is the renal vein. And again, we'll talk more about the branches of the vasculature when we look at the anatomy in lab. So let's start off by looking at the anatomy of the nephron. Okay. So we're talking about the nephron. Just to describe this briefly here, the nephron is pretty much the structural and functional unit of the kidneys. Meaning that the functions that the kidneys carry out take place at the level of the nephron. And there are several, many nephrons that make up the kidney, but the kidney function happens down at the level of a single nephron. Again, we want to distinguish our two regions of the kidneys, so this makes a little bit more sense. We've got the cortex, which is this outer paler staining region, and then we've got the medulla, which is closer to the center of the kidneys. Let's talk about the vasculature here. So we've got an afferent arteriole, which is coming towards the first part of the nephron. This is bringing fresh oxygenated blood. And then we've got the efferent arteriole, which is taking deoxygenated blood away. Right, so this is an afferent arteriole. And this is an efferent arteriole with an E. Alrighty. Now, the blood that's being brought is actually being brought to the glomerulus. So the glomerulus is the first part of the nephron, and the glomerulus is really a ball of capillaries. So it's this ball of capillaries that has been stuffed down into this double-walled membrane. Okay, I'll say that again. It's a ball of capillaries that has been stuffed into this double-walled membrane. So these capillaries, this afferent arteriole and this efferent arteriole, kind of modify themselves and shove themselves down into this double-walled membrane. And this ball of capillaries is what we call the glomerulus. Okay. Now, coming in close contact with the glomerulus is the double-walled membrane, which is the first part of the tubular structures of the nephron. So we've got this double-walled membrane. And when I say double-walled, think about like the peritoneum, think about the pleural cavity. We talked about the pleural cavity, which has two membranes, one that's sticking to the chest wall and one that's sticking to the lungs. There's a very similar thing happening here with the Bowman's capsule. So we've got a parietal layer, a visceral layer that is continuous with this outer parietal layer. Okay. And together, this entire double-walled structure is called the Bowman's capsule. Okay, the Bowman's capsule. Now, the rest of this tubule modifies itself to give us the remaining tubular structures. So we've got this proximal convoluted tubule, which then becomes the loop of Henle, which then becomes the distal convoluted tubule, which finally leads to the collecting duct. Okay. All right, so let's label these out here a little bit. We've got the proximal convoluted tubule. And convoluted just means that it's kind of windy or bendy. And we can see that that is the shape of this part of the tubule, right? It's kind of windy or bendy. We've got the distal convoluted tubule. And then we've got the loop of Henle. And as we can see here, the loop of Henle dips down into the medulla. That's going to be a really important relationship to understand in terms of the function of the loop of Henle, the fact that it dips down into the medullary portion of the kidney. And then finally, we've got the collecting duct over here. And the collecting duct basically leads to the calyces, which leads to the renal pelvis. And then that is where urine can be drained into the ureters. Okay, so this is the collecting duct. So this is the structure of a nephron. And so it's really important to understand this structure as a backdrop for everything we're going to talk about. We're going to talk about how blood is leaving the capillaries here. It's going to be filtered across the basement membrane, the filtrating membrane here. So basically, we're going to squeeze this blood across the glomerulus and into the Bowman's capsule. We're going to speak about the structure of this membrane, this filtration membrane, that allows some particles through and some particles not, right? Some particles are going to get through into the Bowman's capsule, and larger particles like proteins and erythrocytes are going to remain here in the capillaries and make their way back to the efferent arteriole. And that's what we mean by filtering. We'll talk a lot about that. From there, we're going to talk about how the solution or the substance that is now sitting in the Bowman's capsule is going to be fine-tuned as it goes through the various parts of the tubular structures to become urine. And then when it gets to the end of these tubular structures, it's going to be passed down to the ureters and then to the bladder where it's stored until it's time to be voided. Okay, so all of this is to say, how do we take blood and make urine from that blood and eliminate things that the body needs to get rid of, meanwhile reabsorbing or reclaiming things that the body does not want to get rid of or the body needs. We're also going to speak a lot about how the different parts of the tubule, how the cells here look very different from each other, and because they look different, they're going to bring about a different function. This is that form follows function theme that we've been talking about quite a lot for the entire course of this semester. So the cells of the proximal tubule look different to the cells of the distal tubule, look different to the cells of the loop of Henle. And that's because different processes are being are taking place at these different parts of the nephron. Okay, so let's go back here. All right, let's talk a little bit about the vasculature because a lot of what we'll talk about requires you to understand this relationship, how the vessels here are coming into contact with the different parts of the nephron. So just to review what we just talked about, we talked about the nephron. So the structure in yellow here is the nephron. We've got the glomerulus here, which is the ball of capillaries. We've got the Bowman's capsule, which is closely surrounding that ball of capillaries. We've got the proximal convoluted tubule here. And you can see how it's kind of windy or bendy. We've got the loop of Henle. We've got the distal convoluted tubule here again. You can see this is very windy or bendy. And then we've got the collecting duct. And we can see that the loop of Henle dips down into the medulla. Now, the afferent and efferent arterioles are shown here. The afferent arterioles coming into the glomerulus, and then the efferent arterioles coming out of the glomerulus. Now, when the efferent arteriole leaves the glomerulus, it's going to go and wrap itself around the proximal and distal convoluted tubule. These are what we call the peritubular capillaries. So as the efferent arteriole leaves the glomerulus, it's going to intimately wind or wrap itself around the proximal and distal convoluted tubule, and this is called the peritubular capillary. This is really important to understanding how reabsorption takes place. So we're going to filter blood across the glomerulus and into the Bowman's capsule, but there are certain things that are going to end up in that tubule that we want to reclaim. For example, glucose, for example, amino acids. We don't want to get rid of those in urine. And so having these capillaries wrap themselves around these two parts of the tubule allows for the reabsorption of these substances from these parts of the tubule back into the capillary so that the body can reclaim those vital substances. Another important vasculature or a modification of the vasculature here is called the vasa recta. And the vasa recta is the modification of the veins of the kidneys that wrap themselves around the loop of Henle. So whereas the peritubular capillaries wrap themselves around the proximal and distal convoluted tubules, the vasa recta intimately wraps itself around the loop of Henle, okay? These are two relationships that we're going to call on again, so it's really important for you to have this kind of visual or anatomical relationship in your head. Vasa recta intimately connected to loop of Henle, peritubular capillaries intimately connected with the proximal and distal convoluted tubules, okay? All right, so let's begin to speak about the basic processes of renal exchange. So there are three processes involved in renal exchange. There's glomerular filtration, This is where blood or plasma is filtered from the glomerulus into the Bowman's capsule. There is reabsorption. This is where useful, important, nutritive substances are reclaimed from the tubule back into the peritubular capillaries. And then there's secretion. This is where substances that the body does not need can be moved in the opposite direction from the peritubular capillaries back into the capillaries, back into the tubule, excuse me. And then finally, there's excretion, which is the final part of this, where the final product, which is now called urine, is moved from the tubules and out of the body via the ureters and the bladder, etc. So filtration takes place here at the level of the glomerulus. We'll talk about the makeup of the filtration barrier here, which explains why some substances are filtered through and others are not. Reabsorption takes place along the length of the tubules. So important substances can be reclaimed back into the peritubular capillaries, and that is to prevent them from being excreted with the urine. Secretion is where substances that are not needed, this is usually excess potassium, toxins, drugs, medications that we may be taking, can be removed from the peritubular capillaries and into the tubules to be eliminated or excreted. And then the final part of this is excretion, where the final product at this point, which is called urine, is moved into the ureters, moved to the bladder to be eliminated from the body. Okay. Okay, so here's kind of a close-up of that again, and we'll talk about each of these processes individually. So glomerular filtration is movement of protein-free plasma from the glomerulus into the Bowman's capsule. And just to remind ourselves of the difference between the glomerulus, which is the ball of capillaries here, and the Bowman's capsule, which is the first part of the tubular structure, which is this double-walled capsule that is intimately wrapped around the glomerulus. So blood that leaves, and I'm being kind of loose in calling it blood, but it's not actually blood because the erythrocytes do not make it through. So erythrocytes are too large to pass through the filtration membrane. And so what we have being filtered through is protein-free plasma. So the protein stays behind because it's too large, and the erythrocytes stay behind again because they're too large. Now our GFR is about 125 milliliters per minute, or 180 liters per day. And what that means is that of our blood volume, 125 milliliters is being filtered across the filtration membrane or the glomerular membrane every minute. And that adds up to about 100 liters being filtered every day. So the kidneys are hard at work, right? They're filtering large volumes of our blood, of our plasma, every minute and every day in order to constantly clear our blood of excess toxins, clear our blood of excess byproducts, so breakdown products of metabolic processes, excess nitrogen from some of the protein that we eat. All of that needs to be cleared from the body, and that happens across the glomerular membrane to start, okay? Let's talk about the structure of the glomerular membrane or the filtration membrane. So there are three important layers to this membrane. There's the fenestrated capillaries. And we should have talked about the different types of capillaries, but basically fenestrated capillaries are capillaries that have large holes in them or large slits in them. So as we can see here, one of these fenestrated capillaries, we can see that it kind of looks like a sieve in that it has all of these tiny holes in the capillary, in the wall of the capillary rather, and that is allowing some products through and not other products. The second layer of the glomerular membrane is the glomerular basement membrane. So if we look even closer here, we can see the basement membrane of the epithelial cells of the Bowman's capsule. right? So here is our capillary endothelial cell. Here is the basement membrane of the Bowman's capsule. And then here is the epithelial cell of the Bowman's capsule on the other side. So this is the side of the glomerulus. This is the side of the Bowman's capsule. This is the cell that makes up the Bowman's capsule. It's an epithelial cell. This is the cell that makes up the capillary. It's an endothelial cell. So the endothelial cell, the basement membrane of the epithelial cell, and then the epithelial cell. These are the three layers that make up the glomerular membrane or the filtration membrane. Now I want to talk here a little bit about the structure of the Bowman's capsule, the structure of the epithelial cells down here. So these are described as having these foot processes because of these podocytes. So the podocytes are the cells that line the visceral layer of the Bowman's capsule. And they have these large slits in them that we call filtration slits. And this is another modification that allows some substances to pass through this membrane and other substances to remain behind and stay behind in the capillary. Okay. So basically the action of squeezing blood through the endothelial cell, across the basement membrane, and through the filtration slits of these podocytes is what squeezes the plasma from the capillaries and into the Bowman's capsule. And that's the first part of this process called filtration. So here are some other views of that barrier, of that filtration membrane. And just to kind of reiterate here, we've got the Bowman's capsule. It has a parietal layer, and then it has an intimate layer. Now the intimate layer is very tightly adhered, the inner layer, sorry, or the visceral layer is very intimately adhered to the glomerulus. And we can see if we look a little bit closer, the podocytes that make up that visceral layer, and then the slits in between the foot processes of those podocytes that really make up the filtration slits that allow for certain particles to pass through and other particles not. This is what we call a semi-permeable or a selectively permeable membrane. And then if we get even closer here, we can see those three structures that I pointed out. So the capillary endothelium, these are the cells that are lining the capillary, the basement membrane of the epithelial cells, and then the actual epithelial cells themselves, which have these podocytes and the slit processes, or the foot processes.